the slides.
Yep, that's great, Smitha. Thanks.
Thank you so much. I was enjoying your talk so much, Wendy. I forgot that I had to speak. I was very relaxed, and then I suddenly realized I better, get into speaker mode. Thank you all so much for having me. Thank you, Leanne, for this very kind invitation. I just want to acknowledge that I am on Gadigal land. I'm coming from Sydney, and it's a real privilege to be able to talk about this amongst like-minded people. Wendy has really set up my presentation really well because she's highlighted all of the things that underpin my research interest, which is really looking at the basic science part of antimicrobial resistance, how it's sort of the building block, if you will, is probably a good way to describe it, of antimicrobial resistance. And that's what I will be talking about this morning, and hopefully we'll stay on time. So you've had the clinical presentation, and of course you can't see the dentist's face here because, as with the Oral-B ad, we try and keep that with all the cartoons. It's very hard to find a dentist that was smiling. Very happy patient, which is also quite rare because nobody's really that happy to see the dentist, we know. But I would say they're very happy because they've had their teeth sorted, and it's been a preventive appointment. They haven't needed to have anything done, and we've avoided the need for antibiotics altogether, which is, of course, the ultimate aim. But my research focus is, while that's what I do, is seeing patients, and I, of course, part of that is I do have to prescribe antibiotics once in a while, unfortunately. I really am looking at the genetic basis of antimicrobial resistance, and the figure that you can see there is actually an antimicrobial resistance gene, an ARG, and you can see that it's just basically, it's a string of letters. It's the 4 letters that make up our life on this planet, and it's the same 4 letters that are strung together within a bacteria that will spread antimicrobial resistance. And you can see here on this particular streptococcus, so this gene has come from a streptococcus bacteria, so this is the species. There are 2 arrows, and they are pointing to the parts of this genetic sequence which actually are identified as antimicrobial resistance genes. So there's URM-B here, and this is tetracycline, this is macrolide resistance, URM-B, and there's TET-M, which is tetracycline resistant. So one of the things you'll notice straight away is that this necklace of genetic material contains actually 3 different types of antibiotic resistance genes. So scary straight away to think, why are three of them there? But that's the thing. We think about antimicrobial resistance very much in a clinical format. We don't actually think about how is it being spread, what is actually happening at this, very much at this genetic level, and a lot is happening at the genetic level. And the issue that we have is we don't have enough information about it. So we're always playing catch up, because of course, one of the things that we do know is that bacteria are able to evolve frequently and easily. And that always means that we're, it's a bit like being involved in trying to catch drug cheats in the Olympics, obviously on a different level. That's probably not the best example, but they're always behind because, people spend so much time figuring out how to cheat rather than, perhaps doing the training. It's the same with students and exams. But anyway, let me return to antibiotic resistance. So one of the things that I think we forget is we are in this war. That's what, definitely from a clinical point of view, it's always seen as we're in this war with bacteria and we have to fight bacteria and we're going to win. And one of the beautiful things about that infogram that Wendy put up there, where dental disease obviously was deadly in the 17th century. It's hard to think about that now in the 21st century, especially in the countries that we live in. But the truth is, for most of our, we've been on this planet for a blink of an eye. For most of the time, this planet has been, very much dominated by bacteria. And while we've moved on to become, the apex species of this planet in many, many ways, bacteria have been dominating for billions of years. And in that time, they have got the best available defense mechanism in the world. Essentially, they are able to have an amazing ability for genetic mutation, and they have been able to, because, obviously, these organisms, it's exponential growth, they're able then to figure out what is the strongest, what is the best, what will go on to help them. That's quickly identified, and then that is maintained to make sure that that particular species is going to survive, and it has survived. They've survived through everything, and they continue to survive and thrive on our planet. And the other thing that we've realized is that in this war that we've had, and we thought we'd won the war against infectious diseases, antibiotics gave us this new hope, until it showed its ugly side, is that we have now realized, we also can't survive without bacteria. But it's a much more complex relationship than enemy or friend. Or we're coming to this understanding that actually perhaps we need bacteria to live. And certainly in terms of cellular mass in a human body, we are actually more bacteria than human genetic material. So it's an interesting thing when we actually come down to the basics and think about it, because we spend so much time, we're washing our hands, we're cleaning, we're doing all these things, and all of it underpins this paradigm that bacteria are bad for us. We have to get rid of them. And that has fed into philosophy about how we fight bacteria, and also about antimicrobial resistance, because it always is, okay, we need to outdo bacteria. We're going to create a new antibiotic that is going to be better. It's going to do a better job at killing or stopping the bacteria. And the truth is, the bacteria are going to find a way to overcome that. And it's because this is what they've been built to do. They've been built to fight each other and to make sure they survive. And they've had billions of years of a head start than we will ever have. One of the reasons why we have such a better understanding of antimicrobial resistance and this genetic level of antimicrobial resistance is because of the evolution of microbiology. And Wendy touched on this when she talked about dental infections being polymicrobial. 50% of bacteria that is part of the oral microbiome actually can't be cultivated on an agar plate. So, for years, we were living in this, we worked under the notion that, there was probably about 300, 400 species that we'd find in the mouth. And with the evolution of sequencing, so with Sanger sequencing, with 16S sequencing, we'll say there's about 700 species in the mouth. And as we move on to next generation sequencing, so that's what we're currently in, we're in our third generation, we're at long read sequencing now. We are able to understand that, in the mouth, there's, probably 1,000 species, and we can also look at subspecies. Our understanding of microbial communities have been broadened to the extent that we have too much information and we don't know what to do with all of this information. So we've really gone to the other extreme, but it has certainly been technology and this revolution in understanding what microbial communities look like, the challenges involved with dealing with biofilm infections, has really led to this deeper understanding of antimicrobial resistance. Obviously, there is new terminology associated with this. We all know about the microbiome. When we are looking at resistance, we are looking at the resistome. So this is the microbiome, of course, is made up of a number of different types of microbes. Bacteria is one of those microbes, but the resistome comes from the bacterium. It's the genetic elements within the bacteria that will that confer resistance. And we also have the mobilome, which is the mobile aspect of the resistome. So some of those resistance elements will remain within the bacteria. More scarily, the bacteria like to share, and they're very good at sharing and transferring information. And these mobile genetic elements, which carry resistance. And actually that genetic sequence I showed you earlier, it's a mobile genetic element that carry the resistance genes. That's far more worrying, and that's really our area of interest, is that the ability to carry and to transfer resistance. One of the wonders of metagenomics, and I won't spend too much time on talking about next generation sequencing, is that we are, well, when I say we, I think perhaps it's in dentistry, we like to see things, we like to, it's tangible, we can, drill into it, we can cut into it, we can see pus come out of it. It's so terrible to admit to this, but it's absolutely true. It is very satisfying to drain a tooth and to see these things, because you know that you are making a difference. But seeing is believing. A lot of people in this world would say, yes, we are visual learners. So to explain to a patient that, oh yes, if you have this, if we give you antibiotics, it's going to cause resistance. It is an idea. It is hard to fully understand that unless you're somebody that has had a horrific experience and you have survived that. Then it's very tangible to you. But for most people, yes, that there's resistance. I kind of get that. I probably shouldn't take antibiotics. But then, you get sick and it's the easiest thing to do. And maybe I will take some antibiotics because, if on the off chance it might be bacterial. Metagenomics gives us an ability to visualize. And when we say visualize, you are looking at sequences and it's data, but you can see it. It's great when you can see a whole list of resistance genes, and the list goes on, and you think, oh my gosh, that is what you can find in somebody's plaque. But you can also quantify it as well. So this is a sort of a very, very simple graph looking at what antimicrobial resistance genes are found in the mouth. And of course, the mouth has multiple different surfaces, which are quite different. So the saliva, it's planktonic bacteria, but on the teeth, you'll find biofilm. And of course, the teeth are above the gum and below the gum. We've got completely different environments there. And what we were looking for, this is a project done by my students, was how many resistance genes can we find in the last 10 years, looking at next generation sequencing studies. And there's not enough information. But what we did find is that richness, so the number of bacteria, of resistance genes that we could find was most when you have metagenomic studies. Other studies, if you're culturing it, we're not going to find enough. Or if you're doing PCR-based identification, you are not going to find the whole range of these resistance genes. So methodology is also important. And of course, with metagenomics comes a huge bill as well, but that bill is reducing as technology improves. However, to really understand it, we have to have this particular methodology. And when you have this methodology, what you understand is that resistance is really everywhere. These resistance genes, and I was talking about that ability to transfer. So this figure here shows that at the center of the wheel is human consumption that drives antimicrobial resistance and the spread of resistance genes. And then it will, obviously, as we know, it is spread through people. Air travel is a huge issue. We live in a global village. It's very easy to spread infectious diseases. As we've all lived through a pandemic, we know exactly how this happens. Of course, if you're here, we're lucky in the sense we're very far away from everybody. Our borders are very easy to shut down and control. But even then, these resistance genes, they permeate. They will go through land. So you've finished a course of antibiotics, but you've got some leftover tablets. You decide to flush them down the toilet. They enter the waterway. So if you look at resistance gene studies around hospitals, the wastewater has very, very high levels of antimicrobial resistance genes. Even our wastewater treatment plants, they generally have a higher rate. It's in soil. It goes out into the ocean, and it will be in dust particles in our atmosphere, and even in rainfall now they are finding resistance genes coming as they collect the water. So resistance genes are part and parcel of the biosphere because, of course, it's that ancient defense system that I was talking about, but that amount has radically increased, and that is really pushed through because of how we are using antibiotics. And unfortunately, most of that is coming from use in agriculture, animal husbandry, and aquaculture. So this is why One Health is so important. It's very easy to say, oh, okay, if it's, it's happening because of food production, then as a healthcare professional, I don't have to worry. Me giving out one prescription is not going to change the world. But that is the wrong attitude. And I think that was what Wendy was saying. Wendy was saying that as well. Each of us do have an ability to make a difference. And even if it does mean that one prescription, then that is the difference that we can make. And at least it will offset whatever else is happening, which we have less control over. But certainly I feel, I do think it is important that we do advocate for cleaner agriculture. And a lot of countries in the world are doing that. In Australia, our beef industry is very, very heavily monitored in terms of antibiotic use, not so much for chicken, so poultry, and for pork. And I think that there perhaps might be a link there, but leave that with me. And of course, this is my homage. I love fantasy, the genre, and when I was thinking about this, it is the serpent eating itself. There's a eternal pattern to this, but the wheel is turning faster. We are pushing it a little bit harder because of what we're doing. We can sort of push this along, or we can slow it down. We do have that ability. So my PhD was looking at the development of the oral resistome in the first decade of life in a twin population, an Australian twin population, so from 3 months old to about 11 years old. And basically what we found is we took supragingival plaque samples, and they underwent sequence-based metagenomics. And what you can see here is the AMR class. So this is the genes that we found. This were the class of antibiotics they were resistant to, and you can see here. This is time point 1 that we had 3 samples taken along this decade. The samples actually are not evenly spaced out, and the reason for this is it was actually based on dentition. So time point 1 is when it's an edentulous child, so they haven't had their baby teeth come. Time point 2 is when they have their baby teeth. And time point 3 is they're older, is because we're now looking at when they get their adult dentition. So if you wonder why there's a bit of spacing looking at the ages, it's because it actually was related to dentition. But you can see here that there's a lot of blue. There's a lot of genes that are resistant to macrolides. And this was something that was really interesting to me because children are the highest medicated in terms of antibiotics in Australia, and amoxicillin is the #1 antibiotic given to children. So I was expecting a lot more black, which is beta-lactams, but that wasn't the case. The other thing, just to quickly draw your attention to, is this pink area here. This says IS richness. IS is these mobile genetic elements, and richness is the number of mobile genetic elements. And you can see that as we move through the population, as we come to time point 3, there are many more mobile genetic elements found in the oral resistome as children grow older. So this was at 11. It would be wonderful to now look at these children at later time points just to see if this changes, because this is the key here. It's all well and good to have resistance genes in your mouth, but if they're moving around and they have their ability to move increases over time, that's what we're interested in. What we also found was that the resistome, so in terms of its diversity, very much mirrored what the microbiome looked like. So as the microbiome increases in diversity, so does the resistome. But generally in the mouth, things are a pretty even keel. So you'll see big changes. If this was the gut gut microbiome, you'd see much bigger changes. The mouth, things are relatively stable, and they stay stable for a long time. There were a couple of changes sort of early on in life where there was a peak, but then things sort of settle down, and that makes sense because we don't want our mouth to be too reactive. It's there's a lot of external and internal influences, and it's a very dynamic environment. So we would like to make sure that we've got at least a stable microbial base in the mouth. What is the resistome composition shaped by? So I've just explained to you, yes, obviously what the bacteria in the mouth has an impact on this. Because we had twins, we could run a classic twin analysis. So we could look and see if it was host or environment. And often with microbiome studies, this is a little bit difficult because it will always come back to environment. And you can see that E here does, there's a lot of significance for E, but there was some host, the host was also responsible for resistome composition. And this is in specifically looking at a diversity and specific types of resistance genes as well. So there is something there that says, okay, there might be something from, from your own genetic background that could be influencing this. Wendy again had a fantastic graph looking at what antibiotics people were prescribing. Well, this graph takes that information, but then inputs in what we found at a genomic level. So the most prescribed antibiotics in Australia are beta-lactams, it's amoxicillin, but you can see in terms of the contribution, in terms of abundance, in terms of quantity to the resistome, it's actually quite low. Things like, again, macrolides, fluoroquinolones. So macrolides are 20% of the child population would be given macrolide antibiotics, but it really represents a huge amount of the resistome. And fluoroquinolones, which was interesting because they're not prescribed to children in Australia, that you can see quite a lot of fluoroquinolones in a child's resistome. So one of the things that we found was all children had resistance genes in their oral microbiome, whether they were exposed to antibiotics or not. So the genes were there, which we would expect. But those that were given antibiotics, and again, as I said, they were mostly given amoxicillin, that wasn't reflected. We also had maternal samples, so we could look at mum's plaque and look at and see, did what mum had in her mouth, did that have an impact on the child? And M1 is, so this is mum when the baby's 3 months old. And sorry, that's the twins. This is mum when the, so this is at 3 months. And what we could see is there is some similarities. So for instance, if we are looking at, you can see that the vancomycin resistance, there are resistance genes in mum. You're going to notice that it'll be in the children as well. And you'll see as we work through these antibiotic types, there are similarities between mum and the twins at 3 months. When you move to time point 3, where they're 10 or 11 years old, that composition similarity reduces. It reduces quite drastically because obviously children have, they're much more independent, they're doing different things, and their resistance composition looks quite different to mum. We also looked in health and disease, and of course for us disease, the number one disease we fight is dental decay, and there were differences in resistance genes found between the depth of decay as well. And again, that is a reflection most likely of the different microbes involved, the different bacteria involved as the depth of the decay changes. Interestingly, when we looked at teeth that had fillings on them, they had a very similar profile to health. So it had a very similar profile to the resistome in health. So is that a good sign? Again, there are lots more questions that have come out of this than really definitive answers. So why is this even important? This seems like a very, very, very niche area within oral health, which is niche itself. Well, because it demonstrates there's interconnectivity. So the mouth is important because it's the second biggest microbiome in the body. It's also the oral microbiota have the ability to move. So every time you brush your teeth, there is a systemic flushing of oral microbiome. It's moving through our blood system. Are these bacteria carrying resistance genes? It's quite possible that they are, and that's what we really need to do much more work on. But it also shows that within this wide biosphere, there's lots of environmental factors that play a role too, and that's having an influence on what's happening inside our mouth as well. And these are important things to understand, because if we're going to fight antimicrobial resistance, we really need to understand it in its entirety, not just what happens, in a clinic, in a chair or at a bedside. So if we do want to increase the success of AMS programs, and this is certainly from a genetic perspective, I really do think we need surveillance. We need the data to underpin the AMS programs and say, yes, this is what we're finding, and the programs are done, and it's now going to, this is working towards being more effective. So we do need long-term sustainable surveillance at a genomic level. I think that is really, really important to make sure that we are using everything in our armamentarium to fight against AMR. Thank you.
Thank you so much, Smitha, for that fantastic presentation. And it was, really interesting to look at those, all that, the resistome in, twins who, or, babies who have never really received any antibiotics. At this point in time, does anyone have any questions for Wendy or Smitha? Just got probably a couple of minutes. I saw the comment from Ree about the excitement of, draining abscesses in animals as well as humans. We've got a couple of minutes of questions. I just have a really quick question for Smitha. So Smitha, why is it that, or why do you think, because I suppose you instantly know, the reason for having, resistance or in babies having resistance to, tetracyclines, which we don't typically, use in children, fluoroquinolones and macrolides, that was so high for children who've never been exposed to these antibiotics?
So one of the issues with that is the co-carriage. So tetracycline genes are really good at hitching a lift with other genes. So if you have been given a macrolide antibiotic, you're very likely to see tetracycline genes attached to that. That's one of the reasons. This really did shock me because, of course, I had some very fixed ideas about what I was going to find, and of course, never worked out. But I decided to look into this a little bit in more detail, which is why I was talking about the beef industry, but there's people here with far more experience in this. But I do, we looked at what was the protein, what's the most common proteins children eat, and it's chicken and pork, and fluoroquinolones and tetracyclines are given to those animals, so in food production. And so this is an area that I would love to do more research in, because it would be interesting to know if through that chain, is that how it's coming through kids. Oh, so it's not in Australia. Sorry, I'm just looking at the chat there. Oh, okay. Well, that's good to know. Has there ever been a time when they've been given fluoroquinolones?
No, fluoroquinolones have never been registered for use in food-producing animals in Australia.
Okay, but at tetracyclines?
Tetracyclines are, yes.
They are.
They're one of the most commonly used.
Yeah. Okay. That was okay. So that's
yeah.
I didn't
they
realize
have detected fluoroquinolone resistance in poultry despite them never being treated. So I guess it's that One Health.
Yes.
The genes are in the environment.
Yes.
Trying to pinpoint where it's come from. Challenge.
Okay. Oh, well, thank you. Oh, that's really, that is the challenge, isn't it?
Yes.
It's come to 9:30, so right on time. One question. So look, thank you so much to Wendy and to Smitha for speaking today. Smitha's obviously zooming in from Sydney, and Wendy zooming in from Manchester. So yeah, thank you so much everyone for attending, and I hope it was really interesting, something a bit different about dentistry this time for this journal club. Carolyn, did you want to say anything?
Yes, thank you so much. So thank you to Leanne for hosting, and thank you for our presenters today. And I popped in the chat a little bit earlier. Our most recent NCAS circular was actually on the topic of dental infections. So if you're interested to have a bit more of a look, you can have a look there. And also just to say that next month's webinar will be on the topic of primary care. It'll be on Wednesday, the 30th of April, which is a slight difference in our usual schedule. But that'll all be available on the NCAS website. So thank you so much, everybody. Have a great day.
Thank you very much.
Thank you.
Bye.